All right, good evening everyone. So we are here to discuss INICET Recall May 2024 Biochemistry portion. I hope your examination went very well. Most of the topics which we have already discussed so many times under last minute revision topics were from them. Right, some topics here and there, that will always be, but yes, 80 to 90% is the fair game. Anyways, let's start with our questions. And the first question was on, let's discuss the first question on congenital lactic acidosis. Lactic acidosis is very important topic. And the lactic acidosis is due to the defect of which of the following enzymes? Yes, the correct answer is pyruvate dehydrogenase. This is a link reaction between glycolysis and TCA cycle. Take this session as in for those who have just done the examination, right, and are very hopeful for the seat, right, for them, just sit back, relax and see if you are just able to match all your answers and not. And for those who are preparing for the NEET examination, June 1, 2024, please take this as a revision, right? Most of the things we have discussed here and there so many times, but take it as a revision. Whatever comes in the last 2 to 3 years is the most important topic. So let's discuss all of them here, whatever there in the yesterday's paper. So congenital lactic acidosis is due to defect of which of the following enzymes? It is pyruvate dehydrogenase. This is a multi-enzyme complex protein which is made of E1, E2 and E3. The E1 is PDH itself. One of the E enzymes is dihydrolipoyl transacylase. The other E enzyme is dihydrolipoyl dehydrogenase. What is most important is that this enzyme requires 5 cofactors, and I always use the mnemonic to remember those 5 cofactors. That is tender, love, care for Nancy. And I know most of you know it. And this is thymine, lipoic acid, coenzyme A, FAD, NAD. Tender, love, care for Nancy. That should help you remember the 5 coenzymes or cofactors for the enzyme PDH. And of course, any enzyme requiring vitamin itself is an important question. For example, this enzyme here, thymine, is the most important vitamin for this enzyme, and if this is deficient, it results in Wernicke-Korsakoff psychosis. There was another question on Wernicke-Korsakoff psychosis. We shall be discussing it in a while. But another important thing is that there are 3 enzymes which work in the same fashion. That is, the 3 are multi-enzyme complexes made of E1, E2, E3 requiring 5 cofactors, that is thymine, lipoic acid, coenzyme A, FAD, NAD. I'll be repetitive so that you revise that so many times with me. These are the most important topics. So this here includes PDH, yes, number 1. Number 2, there is an enzyme in TCA cycle. If you can recollect, it is alpha ketoglutarate dehydrogenase. And number 3 is this enzyme, branch chain ketoacid dehydrogenase. This enzyme, if defective, results in one of the disorders. Can you name it in the chat box? If this enzyme, branch chain ketoacid dehydrogenase, is defective, what will it result in? It will result in, yes, maple syrup urine disease. It results in maple syrup urine disease. So this is very important. Then the next question. Now there are 2 most important topics. One is von Gerk's disease, and I am not very sure if there was any question on sphingolipidosis. If you could find any question or recollect any question on sphingolipidosis, you can put it in the chat box. Anyhow, those are the 2 most important topics, and put make a chart or make a table for this, or take a print of it, screenshot of this, and please paste it somewhere in your room and do revise it just before the examination. This glycogen storage diseases and sphingolipidosis, those are the two very, very, very important slides in biochemistry. So yes, von Gerk's disease, it is glucose 6-phosphatase deficiency. Pompey's is lysosomal acid maltase defect. Fowlby's disease or Cory's is amylose 1,6-glucosidase defect, which is also known as, this is Cory's, and Cory's is a defect in D-branching. Cory's is a defect in D-branching enzyme. Anderson's is a defect in the branching enzyme, and McArdle's is a defect in myophosphorylase. I always tell this. Two most important topics, sphingolipidosis and von Gerk's disease, and glycogen storage diseases. Out of all these, yes, of course, von Gerk's is very important, but any of them can be asked. Easy way to remember, von Gerk's is type 1, so remember it as von. Pompey's, the cause of death is heart failure. These children usually die within 6 months to 18 months of life. Cory's, C and D, that is Cory's is D branching defect. Anderson's is A, it is branching enzyme defect. McArdle is muscle phosphorylase, MM. These are the easy ways to remember. And based on this, now you can match the following. Yes, Pompey's is here resulting in the Cory's is resulting in the, von Gerk's is resulting in the, McArdle's is resulting in the, this one matches directly. Cory's is the accumulation because that is a defect in the D branching enzyme. So there will be accumulation in the branched product. And von Gerk's disease is hepatomegaly and splenomegaly. Pompey's is a defect in the accumulation of glycogen in lysosome. Pompey's is the only disorder which is a lysosomal storage disease. Only glycogen storage disease which results in lysosomal storage disease. Another important point here is, here in this it is directly, I somehow matched it already for you, but in the examination it was somewhere up and below, and this was not the order. So that is why I was just trying to match the following, but it is already matched for you. But in the examination it was not in the same sequence. However, you have to know everything about these disorders, glycogen storage disorders, everything, very important. Then, which of the following is not a product of heme catabolism? The iron, yes it is. Biliverdin, yes it is. Carbon monoxide, yes it is. See, the problem with aminolevulinic acid here is that we have seen aminolevulinic acid as in the heme synthesis, and hence it is related to heme synthesis but not in catabolism. The glycine and succinyl-CoA reacts with, in the presence of vitamin B6, in the presence of the enzyme ALA synthase to form ALA, and that ALA is aminolevulinic acid. So please note, these are all the degradation steps or catabolic steps, whether you get the heme from hemoglobin or from heme proteins. It will be acted upon by the enzyme heme oxygenase. And yes, of course, it is the next step is a cytochrome P450 reductase enzyme. And this enzyme requires NADPH, which releases iron, which is always in a ferrous form. Then carbon monoxide is released. These are the few reactions in the body where carbon monoxide is released. Usually body does not form carbon monoxide. And biliverdin is formed, which is converted to bilirubin, and bilirubin is then converted to diglucuronide bilirubin, of course via monoglucuronide formation. Now this here is heme degradation. Ultimately it is forming the bilirubin, the bile pigments. So this ALA is present in the heme synthesis, although it is related to heme but is not a heme catabolic intermediate. It's rather an intermediate in the heme synthesis. In the graph given below, what type of inhibitor are A and B? This is one of, I'm so excited about this question because I have so many times said this. Forget everything about KM. Forget everything about Vmax. Even if you do not know any of it, I have always said it. If you see a cross in the graph, that means it is competition and it will be a competitive enzyme inhibitor. If you see a V in the graph, that means victory, no competition. Forget everything else. That is how you will be seeing your answers for, right? Competitive enzyme inhibitor, non-competitive enzyme inhibitor. Of course, for competitive enzyme inhibitor, what will happen to Km? It will increase. The velocity maximum remains the same because it is crossing at the same point on the y-axis. For non-competitive enzyme inhibition, the starting point is same. That means the Km remains the same. However, velocity maximum will decrease. So, always, this is the most important slide under enzymes. The next question. Which genetic defect causes familial hypercholesterolemia? This runs in families, and most common of all this familial hyperlipoproteinemias is familial hypercholesterolemia. This is type 2 familial hyperlipoproteinemia according to the Fredrickson's classification. Also, the type 2 is most common worldwide, and the defect is there is a receptor known as LDL receptor ApoB100E. Its complete name is ApoB100E. Now, I want to emphasize this point. That means this receptor is having two domains. One domain is where ApoB100 is going to bind as a ligand. Another domain is where ApoE is going to bind as a ligand. It is the defect only in B100. What I mean to say is, it is possible that in the options, they gave option A as ApoE, option B as ApoC2, option C, if it was ApoB100, that is my answer. And D can be ApoB48 like this, right? Yes, there can be some variation in the questions. There can be some variation in the options, but take it as an opportunity to revise for the NEET examination. And of course, this is by and large based on the recall from all of you. So, by and large, it is going to be this only. So, which diagnostic tool is preferred for metabolic disease screening in children? The answer is tandem mass spectrometry. I always say this, the most important topic under protein is aromatic amino acids. There are 3 aromatic amino acids. We have phenylalanine, tyrosine, tryptophan. In our notes, wherever we have written phenylketonuria, under that we have written that inborn error of metabolism. The inborn error of metabolism, the screening test is a heel prick test, which is ultimately based on the tandem mass spectrometry. So what they do is basically from the newborn, from the heel of the newborn, they will prick the heel of the newborn and take one drop of blood, not even drop, it's just a dot blot. It is also known as blot test. So you'll just take on the blotting sheet, you take that drop of the blood And with that small drop of blood, we can detect as many as 150 inborn errors of metabolism, even slightly more than that. So approximately these many inborn errors of metabolism can be detected. As the name suggests, tandem mass spectrometry, what it does is, it is detecting the compounds on the basis of their molecular weight. So let's take the example of phenylketonuria. If somebody is having phenylketonuria, in that patient there will be increase in the levels of phenyllactate, phenylpyruvate, phenylacetate. And these can be detected because their molecular weights will be different from any other molecular weight of any other compound. So we detect these compounds like phenylalanine, for example, its molecular weight will be different. So we detect these compounds in the blood. If their concentration is coming out to be higher than the normal individual, that means the patient is having phenylketonuria. Like that, you can detect any inborn error of metabolism using tandem mass spectrometry. For other tests, screening test for the phenylketonuria is, there is one screening test known as Guthrie test. Guthrie test is basically based on a bacterium known as Bacillus subtilis. This bacteria thrives on phenylalanine. So if it is thriving, showing colonies, that means the bacteria is eating on phenylalanine and is happy and growing. And then there is a ferric chloride test. The ferric chloride test is detecting ketone in the blood. And in phenylketonuria, the ketone is phenylpyruvate. And that is the reason we call it ketosis. Do not go for genetic testing. The answer is tandem mass spectrometry. Most common wrong answer will be genetic testing, I'm sure. The metabolic diseases for screening in India is not done for all the newborns, but in US population it is done for all of them. And the rate of inborn errors of metabolism is also higher in US population. Probably that is the reason it is available. TMS is available in India, but we do not use it for all the newborns. And next question is also based on the aromatic amino acids. Which of the following is not an amino acid derived neurotransmitter? Let's see one by one. The dopamine, it comes from tyrosine. Serotonin is 5-hydroxytryptamine and it comes from tryptophan. The GABA is coming from glutamate. We get GABA from glutamate and we get creatine from 3 amino acids. I use a mnemonic to remember this. G is glycine, A is arginine, M is methionine. So this creatine is actually derived from the amino acids, but it is not a neurotransmitter, and hence the answer is creatine. Another question. In folate deficiency, which of the following statement is true? Please note, there can be slight variation in the options based on the recall. However, the concept is important. B9, B12, very important. Vitamin K and the water soluble vitamins, very important. However, out of all the water soluble vitamins, B1, B6, B9, B12, these are the most important ones. Then in folate deficiency, which of the following statement is true? Let's see. Will there be low homocysteine and high methyl malonic acid? What about methyl malonic acid? This increases in B12 deficiency. This increases in B12 deficiency. Also, the homocysteine will be converted to methionine in presence of B9 and B12. Hence, this low homocysteine is not a possibility. The next option is true. This high homocysteine and low methyl malonic acid is true. Homolytic anemia is not a feature for the folate deficiency. The number one cause of hemolytic anemia under enzyme defects is G6PD deficiency. Number two cause for the hemolytic anemia is the pyruvate kinase deficiency. Number three cause of hemolytic anemia is aldolase A deficiency. So there are enzyme defects responsible for hemolytic anemia. Number one, G6PD. Another important point, do not confuse G6PD, which stands for glucose 6-phosphate dehydrogenase, with G6PAs. Which defect was glucose 6-phosphatase? Tell me in the chat box. Meanwhile, let's pen down the hemolytic anemia causes under enzymes. Pyruvate kinase. Then we have aldolase A. Then we have PFK1 defect. These are the enzyme defects which result in hemolytic anemia. B12 supplementation is recommended with folate. Yes, both of them are correct. But if I have to pick between B and D, I'll go for B, depending upon what the exact question was. If the B was not in the options, there is some other options, then depending upon what that option was, the answer can be D. B12 supplementation is recommended along with folate if you see megaloblastic anemia. If the question said, what is the first line of approach when you see megaloblastic anemia? We go for both B9 and B12 supplementation. Another question, which of the following is not associated with post-transcriptional modification? Please note, two very important topics, post-transcriptional, post-translational, both very important. Methylation, there occurs something known as capping, and which is 7-methyl guanosine cap, and hence methylation is a post-transcriptional modification. Then there is something known as tailing, right? This capping is, of course, that is same as 7-methyl guanosine cap, and that is, of course, occurs at the 5' untranslated region. This occurs at the 5' untranslated region of the mRNA. At the 3' untranslated region, there occurs something known as tailing, which is addition of AAAA, also AAA codes for lysine. So it is polylysine tailing. Polylysine tailing. Endonuclease cleavage means there is splicing, and splicing occurs by an RNA known as snRNA, small nuclear RNA, or also known as spliceosome. Please note, we have capping, splicing and tailing. Those are the three post-transcriptional modification, hence is not your answer. Your answer is glycosylation, which is post-translational modification. Biochemistry is not difficult topic. What they do is they play with words. They confuse you between similar terminologies. Post-transcriptional, post-translational modification. Other post-translational modifications can be, there can be ADP ribosylation, methylation, but in this case it will be methylation of proteins, because in the option A it is not specifically mentioned. We'll take it as capping, portion of capping, because glycosylation can never be a post-transcriptional modification. It's definitely a post-translational modification. Then acetylation, ubiquitinylation, addition of ubiquitin. It's a small protein designed for making the protein to die. Then we have sumoylation, of course glycosylation, citrulination, addition of citrulline, addition of sumo proteins. All these are protein modifications, hence it is post-translational modifications. Then another question. This is related to what we have seen. The first question that was on the PDH complex. That enzyme requires thiamine, vitamin B1. And if vitamin B1 is deficient, chronically it results in Korsakoff psychosis. The Korsakoff psychosis is characterized by amnesia, confabulation, and it never causes ophthalmoplegia. It never is causing ophthalmoplegia. Yes, this is seen together. It can be Wernicke-Korsakoff psychosis, acute deficiency of thiamine. That is why I will go with 3 and 4 not being seen in the Korsakoff psychosis. That is why my answer is D. Depending upon what was asked, what were the options, right? It was actually, we have discussed so many times. You remember Goa for Wernicke-Korsakoff psychosis, and under Wernicke-Korsakoff psychosis, there is Wernicke's and Korsakoff psychosis. Korsakoff psychosis is amnesia, it is confabulation portion, right? Neuropathy is overlapping, ophthalmoplegia is not seen. Ophthalmoplegia is a feature of Wernicke's psychosis, right? Another question. This was a slightly trickier one, but still, if you see the segment on genetics, if you have a knowledge about genetics, most of the students don't like genetics portion, I know, but yes, this is based on a part of genetics portion, right? And that is because most of the students do not study this in the INICET examination. It is almost always there. In the last examination on INICET, genetics was part of it. In this examination also, there are 2-3 questions on it. So which type of mutation results in the reversal to the wild type of phenotype when the mutant gene is suppressed? This is a suppressor mutation. You know point mutation, right? Point mutation means there is just one nucleotide defect, right? Now let's see another terminologies known as back mutation and suppressor mutation. I'll be focusing mainly on suppressor mutation because the question is suppressor mutation. Let's see. A suppressor mutation means there was one mutation that has occurred. A second mutation occurred on top of the first mutation, which can alleviate or it can revert the phenotypic effects of already existing mutation. Whatever is already existing mutation, it can reverse its effects. That means no mutation, back to normal. It is a reverse mutation. Yes, it is reversing the effects of the previous mutation. It masks the effect of the starting mutation, and it will not occur at the same point in the DNA. Now, this is slightly trickier. Let's see what I mean to say. Let's say there is a defect in the DNA. The DNA has always mutation is a defect in the DNA. The DNA formed the mRNA. The mutation got carried forward in the mRNA. Now the mRNA is going to form the protein. When the protein is going to be formed, you need a tRNA. Now what happened was the tRNA, right, whatever is the mutation in the mRNA, tRNA could not recognize it. Whatever is the original amino acid which was to be incorporated, somehow tRNA incorporated the same amino acid. This is suppressor mutation. That means originally, if this DNA was going to form a protein A, after the mutation, the protein A is formed again, right? So there were two-step mutation, one occurring at the level of DNA, another occurring at the level of the tRNA. So it is a mutation of tRNA. Yes, this is a slightly newer concept for most of you, right? But if you could have seen this with the keys, then it might have been little easier. Let's see, because the frame shift mutation, it will change the frame. It has nothing to do with suppression. Like, at least we can see the English words and then we can code what the question is trying to say. Addition of another normal gene, that happens in pseudogenes, that happens with transposons, retrotransposons, that we have discussed. Deletion of the mutant gene. If we delete the mutant gene, then there will be no product formed, right? But in this case, that is saying that there is a reversal of the wild type. Wild is always normal. Mutant is mutant. There are two variants of an allele. Either it will be normal or you can call it wild. And the mutant is called mutant only. So either there will be a wild or a mutant gene. So if I have to convert the wild back to wild, I will need a mutation of the tRNA. I will need the mutation of the tRNA. Alright, then what is fluorescence in situ hybridization used to detect all the following except? FISH is used to detect chromosomal aberrations. It is used for detecting any abnormality in the chromosome. The point mutation is going to defect at the molecular level. It is going to defect at the level of the DNA sequence. So point mutation is your answer. The FISH can detect copy number variations. It can detect translocations, amplifications, gene deletion, anything where there is a macrosomal problem in the DNA, that is detected by FISH. It is a hybridization technique where you will send a probe against the already known segment or the segment which you want to identify. But it is done in situ. You do not take the DNA outside and do the study. You are doing everything inside the cell, and hence it is called fluorescence in situ hybridization. Another question, which of the following deficiency is associated with sexual maturation defect or hypogonadism? The answer is zinc. Zinc is one of the favorite topics under your INICET. The zinc deficiency is also known as acrodermatitis enteropathica. It's a rare inherited disease of abnormal zinc absorption. It's an autosomal recessive disease which is characterized by dermatologic, ophthalmologic, GI and neuropsychiatric features along with growth retardation and hypogonadism. The other features are depressed immune system, and there is a taste sensation is lost and night blindness. If you see this is acrodermatitis enteropathica, diarrhea, dermatitis. Then which of the following is the correct sequence? Which of the following is the correct sequence? Whenever there is a hormone, which is water soluble hormone, this water soluble hormone, what it will do is, it will adenyl cyclase will be activated. It will convert cyclic AMP to ATP to cyclic AMP, activating protein kinase A. This protein kinase A will activate a cyclic AMP response element. So the correct sequence is, we start with the adenyl cyclase, then we start with cyclic AMP and protein kinase A. Both C and D in this case is same. Of course, there will be some different in the original question. And this is one bonus question. I always say it along with the glucose transporters. SGLT2 inhibitor empagliflozin is responsible for UTI because it will cause renal glycosuria, which will result in the increase in it will facilitate the growth of bacteria. These are the last minute revision topics for the NEET examination. Gluconeogenesis is very important for the INICET but is also relatively important of all the carbohydrate metabolism. Aromatic amino acids, do not miss on glucose transporters, glycogen storage diseases, sphingolipidosis and some of the techniques. Thank you. Hope for the best and prepare for the next. Thank you.